Welcome to the webinar series Laboratory Good Practices from Greiner Bio1. My name is Thomas Schreiber and I'm a product manager with Greiner for more than 10 years on cell separation products. This is the first webinar of a webinar series called Laboratory Good Practices, and I'll talk today about density gradient cell isolation. In case of any questions, you'll find our contact information at the end of the presentation. In the webinar, we will address some fundamental concepts for cell separation, followed by parameters that are important to control for successful separation. Finally, an extensive troubleshooting section provides solutions for the most common problems that can arise during separation. Let's start with some basics for cell separation. There are many ways to get access to single components out of a complex mixture of cells. Free-flow electrophoresis is a rather old-fashioned method for the use with cells that was superseded by flow cytometry. Immunological methods include panning and the use of immunomagnetic beads. One of the most powerful methods for genomics, proteomics, and cell biology is the separation of particles by sedimentation using centrifugation. Differential centrifugation makes use of the fact that particles of different sizes in a suspension will sediment at different rates, with larger particles sedimenting faster. By applying a series of increasing centrifugal force cycles, pellets containing cells of decreasing sedimentation rate can be obtained. Drawbacks of this method are the low resolution and poor recoveries. A higher resolution can be obtained with centrifugal elucidation, but the method requires rather sophisticated and specialized instrumentation. Density gradient centrifugation combines the use of standard instrumentation with a method that provides both high resolution and good recoveries. First experiments were done in the 1950s with sucrose solutions for the separation of cell organelles and in the 1960s with cesium chloride gradients for DNA separation. Density gradient centrifugation is based on the movement of spherical particles, such as cells in a fluid. The separation of cells can only occur if different cells have different sedimentation velocities. The relationship between the components in this system is described in the Stokes equation shown here on the right. With a given gravitational force, the sedimentation velocity of a cell depends on the cell diameter or size, the difference in the densities of cell and medium, and the viscosity of the medium. When the densities of the cell and the medium are equal, the sedimentation velocity becomes zero and the cell stops migrating. By the way, please note that density and viscosity are temperature dependent. From the equation, it becomes obvious that cells can either be separated by cell size or by their buoyant densities. Therefore, two different methods are distinguished for density gradient centrifugation: rate zonal centrifugation and isopycnic centrifugation. For rate zonal centrifugation, the separation medium has a lower density than the cells. They are separated by cell size or mass, and larger cells move faster than smaller cells. If one would spin the cells for a very long time, all the cells could be pelleted at the bottom of the tube. Isopycnic centrifugation makes use of a separation medium that has a higher density than cells. Cells are separated according to their density, and in contrast to rate zonal centrifugation, cells settle at their buoyant density, even if centrifugation runs for a very long time. Since the separation is based on density, the osmolality of the medium is important. Shrinking or swelling affects the density of cells and therefore their position in the tube with the separation medium. Separation can be performed with continuous or discontinuous gradients. Continuous gradients are made with a gradient mixer and are better suited for analytical purposes. Discontinuous gradients allow for an easy harvest of cells because they settle at the gradient interface. For the following, only discontinuous gradients will be taken into consideration. One of the most common applications for discontinuous density gradient centrifugation is the separation of blood cells. Blood consists of several cell types such as erythrocytes, granulocytes, mononuclear cells, and thrombocytes. The peripheral blood mononuclear cells are of special interest for many immunological questions. Another application is the isolation of mesenchymal stem cells or hematopoietic stem cells from bone marrow or umbilical cord blood. The initial experiments by Bouillon in the late 1960s with polysaccharides and radiopark material as a separation medium paved the way for one of the most popular applications for cell separation. The method takes advantage of the different densities of blood cells. The separation medium makes the erythrocytes aggregate and causes granulocytes to become hypertonic, which leads to pelleting at the bottom of the tube for both cell types. Mononuclear cells settle at the plasma-medium interface, while platelets can be found in the plasma fraction. The approximate densities of blood cells are displayed here. Although a specific subset of cells is present in a density range, separation media with a defined density of one subset allows for pretty targeted enrichment. For example, separation media with a density of 1.077 gram per mil will give an enriched fraction of mononuclear cells, including lymphocytes and monocytes after separation. When enriching or separating a specific cellular subset out of a complex mixture such as blood, positive selection or negative selection can be done. Performing positive selection with density gradient centrifugation reduces the complexity of the target sample to a considerable amount because mononuclear cells are purified over erythrocytes, granulocytes, and most of the platelets. On top of that, dead lymphoid cells are removed. On the other hand, there's some loss of target cells due to cell-cell interactions or because the density span that is inherent in any cell subset. For negative selection, erythrocytes can be removed by lysis, leaving granulocytes, mononuclear cells, and platelets. The resulting sample will have a higher complexity compared to positive selection, but the yield of the remaining cells might be higher as well. For more sophisticated cell separation, density gradient centrifugation is oftentimes combined with either immunomagnetic bead separation or immunoresetting. The actual separation procedure starts with the collection and dilution of the blood sample. A dilution of 1 part blood sample and 2 parts balanced salt solution is recommended. This sample is layered carefully and slowly onto the separation medium in a tube. Ideally, there should not be much mixing between the two phases. Centrifugation has to take place in a swinging bucket rotor with low acceleration and no brake at 400 times G for 30 minutes at room temperature. As mentioned before, it's very important that all reagents and the centrifugation equipment have room temperature because the density of the gradient is temperature dependent. After centrifugation, the plasma is removed and the enriched cell fraction containing the mononuclear cells is harvested. After spinning the harvested cells down, they are washed 2 times with a balanced salt solution. A typical outcome of the procedure is displayed here. Separated cells should have a high viability and 50 to 80% of the initial mononuclear cell number in the blood sample can be expected. Per mil of blood, these are about 1 to 2 million cells. Purity of mononuclear cells should be very high, with only low numbers of contaminating granulocytes or erythrocytes. A typical result for the distribution of different lymphocytes is shown. A facilitation to the laborious and time-consuming procedure of overlaying the diluted blood sample are leukosept tubes from Grindr Bio 1. The tubes have a porous separation disc that prevents the mixing of blood sample with the separation medium. The basic protocol for separation remains the same, and after centrifugation, red blood cells and granulocytes collect underneath the barrier. The enriched mononuclear cells remain above the barrier and the separation medium. In this way, the barrier also prevents mixing of enriched cells with unwanted cells during harvest. The tubes are available pre-filled and unfilled for any choice of separation medium. For the second part of the presentation, I'd like to highlight some key parameters for the separation procedure. Before separation is started, the following important points should be considered. In general, the blood should be processed as soon as possible because the cell quality deteriorates with increasing time. Reasonable results can be obtained until 24 hours storage time. Several studies suggest to store the blood sample at room temperature. Moreover, the blood sample that is ready to be used has to have room temperature as well as reagents and equipment for best separation results. This is due to the fact that there's an inverse correlation between temperature and density of the separation medium. If the temperature is too high, the density of the medium is lower and mononuclear cells move into the separation medium, leading to a blurry interface that's difficult to harvest. On the other hand, if the temperature is too low, the density of the medium increases, which in turn increases the time needed for separation. In addition, erythrocytes aggregate less, which prevents them from entering the separation medium. As with every cell workflow, but especially if the outcome of an experiment is not as expected, it might be a good idea to have an initial assessment of the cell sample used. Cell number, viability, and identity are good parameters to check, which can be done fairly easily with flow cytometry. A balanced salt solution without magnesium and calcium is recommended to avoid unwanted cell-cell interactions. After separation, the separation medium should be removed immediately with washing steps. First of all, separation media might have a slight cytotoxic effect on sensitive cells, but further, the medium might also affect downstream methods. For example, saccharose-based media can disturb lectin staining. In general, any anticoagulant present in commercially available blood collection tubes can be used, such as EDTA, heparin, citrate, or acid citrate dextrose or citrate phosphate dextrose. If there's a choice, EDTA is preferable because it's compatible with most downstream applications. A literature search might identify the most suitable anticoagulant for a given downstream application. The use of defibrinated blood is not recommended because it has been shown that this might negatively affect the mononuclear cell yield and it might potentially increase red blood cell contamination. There are three main classes of separation media that are mostly used for density gradient cell separation. Common to all of them is that they should be adjusted to an osmolality of 290 to 300 milliosmol, which is typical to most mammalian fluids. Polysaccharides, such as ficol, are made of sucrose that is copolymerized with epichlorhydrin to have an average molecular weight of 400,000. Often an iodinated additive such as diartritoate, which is a radiopaque material, is added to adjust for density and viscosity. In fact, there are several non-ionic radiopaque materials that can be used as a separation medium. These are derivatives of triodobenzoic acid. The third group of separation media are colloidal suspensions of silica particles with a diameter of 15 to 30 nanometers. They have a polyvinyl pyridone or silan coating to prevent interactions with cells and can be easily adjusted with salts or sucrose for the desired osmolality. Most of the separation media can be self-prepared or obtained as ready-made solutions with different densities. For the isolation of lymphocytes and monocytes, 1.077 gram per ml is the density of choice. For lower density monocytes and mesenchymal stem cells from bone marrow, a lower density is suggested for the separation medium, such as 1.073 gram per ml or 1.068 gram per ml. Granulocytes can be separated with a higher density separation medium, such as one with 1.113 gram per ml or 1.119 gram per ml. It's also possible to layer a 1.077 gram per ml medium over 1.119 gram per ml medium for the separation of mononuclear cells and granulocytes in one step. Due to the slightly higher density of lymphocytes in animals, such as rodents, dog or rabbits, the use of separation medium with a density of 1.084 gram per ml or slightly higher is recommended. In case your experiment didn't go as expected, the following troubleshooting section might be helpful. Before starting with your precious sample, it's always good to check some basics with the reagents used, such as the expiry date. Is there some discoloration? Are there precipitates or contaminations visible? Preservatives should be avoided because they might affect the viability of sensitive cells. If fetal bovine serum is used, it should be heat inactivated to avoid any side effects due to bioactive ingredients. Note that most separation media should be stored away from light. After separation, different observations might be made. If low mononuclear cell viability is observed, then potential sources for this problem could be either low initial cell viability, the blood sample might have been too old, which means more than 24 hours old, or sensitive cells might have been used, such as, for example, cord blood cells. Potential solutions include checking the input cells carefully for cell number, cell viability, and so on, using fresh blood only, adding protein to the washing buffer, usually 1 to 2% bovine serum albumin or fetal bovine serum are used, or also another separation medium could be used. For example, if fecal has been used, one could try Percol, for example. A low mononuclear cell yield might be caused by if the blood volume is too low, if the blood is too concentrated, the cells might get lost during separation, centrifugation brake that was used, or if the centrifuge is not properly balanced. If the temperature is too high, then erythrocytes aggregate and trap mononuclear cells. And also the density of the separation medium is lower, which causes mononuclear cells to enter the separation medium. Potential solutions include an increase of the blood volume or the dilution of the blood sample, checking for the initial cell number and also checking the protocol, check on the centrifuge whether there's water in the buckets, which can occasionally occur and can lead to a misbalance in the centrifuge. And lastly, also all reagents and the centrifuge, of course, should be at room temperature. Should the mononuclear cell layer be too thin or blurry, then likely sources are insufficient centrifugation, mixing of the phases, or a hyperlipidemic blood sample. Potential solutions then are to check for the centrifugation settings, a more careful sample handling, or to use a fasting blood sample to avoid a hyperlipidemic one. If the mononuclear cell layer is too close to the pellet, then the hematocrit might have been too low in the blood sample. The blood sample might have been diluted too much, or the volume of the blood might have been too low. So potential solutions are to either lower the dilution of the blood sample or to also use more blood volume per tube. The purity of mononuclear cells after separation is quite high, or should be quite high. If there are contaminating cells, then the troubleshooting is directed towards which of the specific cell types is actually the contaminating cell type. For granulocytes in the mononuclear cell layer, likely sources are that the centrifugation brake was used, or that the centrifuge is not properly balanced, or the presence of granulocytes with pathologically low density might also be one of the reasons. Potential solutions are to follow, of course, the protocol, to check the centrifuge, and also to try a lower density separation medium to achieve a better resolution between these lower density granulocytes and the mononuclear cells. Erythrocytes in the mononuclear cell layer can appear if the temperature is too low, which causes the erythrocytes to aggregate less and the medium to have a greater density. The centrifugation time or the gravitational force in the centrifuge could have been too low. The pathologically low density of erythrocytes could be the case, or if the hematocrit of the blood sample was too high. Potential solutions include using all reagents and centrifuge at room temperature. To follow the protocol, a lower density separation medium might help, as well as hypertonic erythrocyte lysis. And in case of hematocrit being too high, further dilution of the sample will likely help. Thrombocytes in the mononuclear cell layer are likely observed if heparin was used as an anticoagulant, if the blood sample was too old, if there was inefficient removal of the platelets, or if too much plasma or separation medium is still present during the cell band collection. Potential solutions include using another anticoagulant, especially ACD, for example. In this case, an acidification of the diluting buffer to pH 6.5 could help. The platelet aggregation could be stimulated with ADP before the mononuclear cell separation. A reduction of the centrifugation speed at the washing steps to about 60 to 100 times G could help, or the addition of a second centrifugation with 4 to 20% sucrose gradient layered over the actual separation medium. Monocytes and lymphocytes usually have a very similar density, so they will appear side by side in the mononuclear cell layer. If the goal is to only separate or enrich lymphocytes, then the monocytes have to be removed, and this can be done with incubating the cells with carbonyl ion prior to the separation. The monocytes will ingest the carbonyl ion and then have a higher density with the separation procedure, so they appear either on the pellet or in the separation medium, but apart from the actual mononuclear cell layer. The following references are useful for more details on the method and for special applications. An excellent piece of advice are the method reviews provided by the current protocol series. 2 papers concerning blood cells are mentioned here as a reference. The publications by Serumwerk Bernburg and Cytiva provide a wealth of information also on specific applications. Finally, 2 books, one of them only available in German, summarize various methods comprehensively. For further information on Greiner Bio-1 separation products, you can refer to the following publications. Flyer and instruction manual are available for both Leukozep tubes and OncoQuick tubes. OncoQuick is a similar product than Leukozep and facilitates the isolation of circulating tumor cells. Now we reach the end of the presentation, and I'd like to thank you for your attention. In case of any questions, please contact us via email at webinars@de.gbo.com. Stay curious and watch out for other webinars from Greiner Bio-1 Laboratory Good Practices. Take care and goodbye.